Alzheimer's disease is an inherited genetic disorder. It is influenced not only by genetic factors, but also by lifestyle and environmental factors. The risk of Alzheimer's is influenced by multiple genes. Alzheimer's is inherited in an autosomal dominant pattern, meaning that if a parent carries a faulty gene, each child has a 50% chance of inheriting that gene. One gene variant known as APOE on the E4 allele is associated with a higher risk of developing Alzheimer's disease. Two proteins involved in the disease are beta amyloid and tau. Beta amyloid is derived from the breakdown of a larger protein called the amyloid precursor protein. In Alzheimer's, abnormal levels of the beta amyloid clump together outside neurons, forming plaques. These plaques disrupt cell function, which impairs communication between neurons. The other protein, tau, binds to and stabilizes microtubules, which are essential for transporting nutrients from the cell body to other parts of the neuron. In Alzheimer's, chemical changes cause tau to detach from the microtubules and bind to other tau molecules, forming neurofibrillary tangles inside neurons. These tangles block the neuron's transport system, which disrupt communication between neurons and negatively affect synapses, especially in brain regions related to memory.
Around 150 SNPs have an effect on early-onset Alzheimer's disease. There are many variations in SNPs that affect Alzheimer's but are not being tested by 23andMe. For example, the RS145999145 SNP of the PLD3 gene is not tested for by 23andMe but can double the risk of Alzheimer's disease. This gene encodes a protein that influences the processing of amyloid beta precursor proteins. This gene is expressed in neural cells. There are many other SNPs similar to this one that have other drastic effects on the Alzheimer's disease, but are not being tested for by 23andMe.
23andMe tested the APOE, E2, E3, and E4 genetic variation. The E4 variation is the only one that will highly increase the chances of developing Alzheimer's disease. This gene typically makes a protein that carries cholesterol and other types of fat in the bloodstream. It is typically expressed in the brain. One APOE E4 gene will double or triple the risk of developing Alzheimer's disease. However, two APOE E4 genes will increase the risk of developing Alzheimer's by 8 to 12 times. There are other genes such as APP, PSEN1, and PSEN2, which can increase the risk of developing Alzheimer's disease before the age of 65.
To surpass 23andMe in predictive value, our test includes about 1,500 to 2,000 SNPs associated with Alzheimer's risk, compared to the about 600 SNPs tested by 23andMe. Although APOE is a significant predictor for Alzheimer's risk, we are focusing on another gene with a strong association not tested by 23andMe. This gene is called SORL1, and it plays an important role in amyloid precursor protein processing. SORL1 encodes a sorting receptor that helps regulate the pathway of APP within cells. This process is critical because APP can form amyloid beta, which, as we discussed earlier, this beta is a toxic protein that accumulates in Alzheimer's disease. Variants in SORL1 alter how efficiently the protein can transport APP away from pathways that lead to amyloid beta production. To design the SNP chip test, we created a SNP array with probes that target both well-known variants like APOE, E4, and those in SORL1, as well as additional genes such as ABCA7 and BIN1, which also contribute to Alzheimer's risk. By including SORL1 and other variants with significant contributions to Alzheimer's, our SNP chip test offers a more comprehensive genetic profile than 23andMe.
There are many limitations to our 23andMe test, but the main concern is that the results will come back as inaccurate. This may be due to incomplete information. For example, some genetic variants might not be fully understood and able to be interpreted to the best of one's abilities, or there may be a lack of information on symptoms or what one can do to help prevent or treat this condition. There are also two main ethical considerations when talking about wanting to take the 23andMe test, and those are financial and emotional concerns. Being treated for genetic diseases can cause high medical expenses for things such as ongoing care, therapies, or medications. For example, according to the Director of Drug Development Pipeline Research at Tufts Medical Center New Digs Initiative, Colin Young, the average gene therapy is likely to cost at least $1.5 million. Furthermore, the emotional challenges one faces after receiving genetic testing results can vary, but oftentimes lead to mental health challenges. There's a variety of emotions, whether it's on an individual, family, or community level, that can cause this. For example, guilt of passing a genetic disease down to children, or heightened anxiety about your health and future.